Right, I think we're on. Good afternoon, everybody. Happy Tuesday. Welcome to Lockdown Bioinformatics, day number 7. Okay, so let's make a start. Right, yes, where are we? Okay, yesterday, what did we speak about yesterday? Yesterday we talked about some tools for starting to work with data. So rather than just creating and moving files around, we started to talk about what we can actually do with the data in those files once we've got some data. So we started by downloading some data, which I'd prepared for the lesson, and then we used a selection of Linux command line tools for working with that, with said data. So among the tools that we used were, I'm just looking for yesterday's notes, just to the files are counting the bytes, counting the lines, sorry, Noah's distracting me in the comments. Hi, Noah. And so column for presenting our CSV file in a nice, clean, tabular way. And then cut for pulling out particular columns from our tabular data. Some natural ordering of the data that's in the file. So by default, sort works on the beginning of the line. So the beginning of our lines in our files was the ensemble gene IDs. So by default, it would sort our file by alphabetical ensemble gene IDs. But we looked at the configuration of sort and figured out how we could use the options that we can give to sort to, instead of sorting it on just that first column, to sort it on the different columns of the data, and also how to make sure that we sorted it in the right way. So not just doing a sort on alphabetical order, but also being able to do a numerical sort, so should such a thing be suitable for the data that we were looking at. So particularly when we were looking at fold changes and p-values and sorting by those, we saw that using a general numerical sort was better and gave us a proper sorting order rather than an alphabetical sort. So that was kind of the beginning of us working with data. What I wanted to do today is two things. One, I want to look in a little bit more detail at input and output streams and how we redirect those streams to various places. Because I realized that we got relatively in depth with that relatively quickly without me ever really doing a proper explanation of it. So I want to talk a little bit about input streams and output streams and how to redirect them. And then I want to look in a little bit of detail at one particular tool for working with data. That's a tool called grep, which we will look at in some detail, again, because it's one of these tools which can be very, very useful if you know how to use it. So spending some time on. So probably most of today's lesson. Okay, so if my 12-year-old has now left the comments, which it looks like he has, we'll move on with the lessons. He's also, of course, while live streaming me downstairs, is consuming my bandwidth, so making the video considerably worse for the rest of you, I'm sure. So hopefully he's now off, and I've got all of my lovely bandwidth to myself. Okay, let's move on. So input and output streams. That's the first thing that I want to talk about today. So for that, I'm going to do some drawing, I think. So let's get to Jacob's picture of the day nice and early in today's stream. So we'll just switch screens. Here is he's done a nice little message for everybody today. Okay, so input streams and output streams. So I'm going to just move, rather than deleting that picture because I like that picture, I'm just going to move it off the screen. So we'll just move it up like this. Maybe we'll keep the hearts there. Right, okay. So input streams and output streams. So we're going to talk about the standard streams in Bash. So the standard streams are, excuse me, the standard streams are called three things. So you've got std in, which stands for standard in. We've got std out, which stands for standard out. And we've got std which stands for standard error. Most Unix tools, so if we have a tool in a box here, okay, tool, most of our standard tools which we use at our Linux command line will usually operate on a file. So their input, so if we have our input here, that's usually a file. So for example, a simple example is cat file, where the input is a file. Okay, so I've written that in a place you can't see it. There we go. And the tool will produce its output in two places. Okay, so it will produce standard out, and that's its kind of, that's the output that comes out of the tool. Wait while my my whiteboard catches up. It will also produce, tools will also produce output to another place called standard error. Okay, so it's not going to catch up, so let's just quickly refresh. Okay. Fine for bandwidth today. Looks like it's taking longer than expected. Okay, so we get two, there we go, two output streams from our tools, standard output and standard error. The key thing about most of the tools, and the thing that makes this, makes tools really flexible for using output redirection in lots of meaningful ways, is you can change you can change where the input comes from. So as I said, most tools will take input from a file, but they will also take their input from what's called standard in. So this is an input stream. So if we just delete this, so the alternative to standard to using a file is to give the tool standard input. Okay, std in, which you can do in lots of ways. So there are other ways of doing this, but the way we usually do it is by piping into the command. So rather than giving cat a file here, we can instead give it standard input by giving it the output of some command. So you've got some command here which produces its output to standard out, and we can send that to standard in for our tool by using this pipe symbol, this vertical line. And so this is how we begin to build up chains of commands by taking the standard output from one command, so that standard output which is produced by, in this case, this sum command, and sending it to the standard input for the next tool in the chain. And it's the pipe that does that. So that pipe symbol in the middle is the key to moving that data around from one tool to another. So you have, if we just redraw this, so you have a tool at the beginning which produces standard output. That standard output is sent as standard input to another tool. Okay, and in order to do this step in the middle here, this conversion from standard output to standard input, that's what the pipe symbol does. Okay, so that converts the standard output from one tool into the standard input for the next tool. And you can keep going. Okay, so the standard output from your second tool can become the standard input for your third tool, and so on and so on. And at any point, you can redirect this output to a file. So you want, if you get to the end of this chain and you want to keep your output, you don't have to send it to another tool. You can send it out to a file and capture the results on your disk. I'm just going to shift this along. I keep forgetting that moving it on my tablet doesn't move it on your screens. Okay, so that is the basic idea behind output redirection. I also want to mention at this point a tool which I learned about today, and this is the really interesting thing about Linux, is that you can be a long-term user. You can have been using it essentially every day for 15 years, and you can still learn about new tools. And I learned today about a tool called TEE, and T gives you a way of both redirecting your output to a file and sending it on to the next command. So if you do command 1 pipe T file pipe command 2, what happens here is the standard output from command 1, so that's this arrow here, okay, gets sent to command 2, but also, and this is why it's called T, because you get this T-shaped thing in the way that it's drawn, gets sent down here to file. So this is a way of sending your standard output both onto another command, but also out into a file at the same time. So you don't have to stop your chain and stop and save the intermediate results and then carry on, and so on. So that's quite a kind of neat tool, which as I said, I only learned about today while I was looking for a way of visualizing this output redirection. So I don't know if that makes it any clearer for anybody. I hope that there's some sort of idea here of how you can move the kind of output from tools from one tool to another, and this process of this idea of transforming the standard output from one tool into the standard input for another tool using the pipe symbol. And I hope that that makes it a little bit clearer. If you really want to break it down, you can do. So if we let me just remove this. You can do. You could do tool to file, then tool 2 on file to file 2, then so if this is tool 2 and this is tool 3 here, then tool 3 on file 2 to file 3 and so on. So it is possible to break it down like one process at a time and save all of your intermediate results to files and do it that way and build up your pipeline by making sure that you save all your kind of intermediate results, which is quite a kind of nice way of doing things if you're working on a fairly small tool chain and on a fairly small set of data. But as soon as you start to work on kind of genomic scale data where your intermediate files may be gigabytes in size, then you end up having to write lots and lots of data to the disk, which in itself is a very slow process. Memory, RAM is much faster than spinning disk or even SSD. So you're doing things in pipes can be much more efficient in the long run. Okay, so that's output redirection and the different kind of input and output streams that exist in Linux. And hopefully that's made that a little bit clearer to people. What I want to talk about now is the one tool that I'm going to introduce today. So I'm going to spend the rest of today talking about one single command line tool, and that command line tool is called grep. Grep is an extremely useful and versatile program, and the idea behind it is that you use it to search for and return matches to particular strings in a file. So I'll try and illustrate what I mean by way of example, and then we'll go from there. So, okay, grep. Let me just find where I'm at. Right, grep. Right, okay, let me open my VM. So here's my virtual machine. I've just left it where I was at yesterday. So so you can see what we were doing yesterday with the sorting commands at the command line. So using sort to sort on the p-value. Okay. I see there's some discussion about this in the comments and whether this is working properly for people. But I'm just wondering, I'm just trying to look at and see what the issue is to see. So the person who's having the issue is seeing that sorting on the adjusted p-value isn't working correctly because we think because of the e expressions in the p-values. So this does work for me as demonstrated here. I'd be interested to know in what way it's not working. So is it sorting alphabetically, or is it sorting, what is it that's wrong about the sorting, essentially? If you're doing the command as I have it here, and let me just go back to that in my history. So this is the command without the less. So that's the sorting command that sorts on the 7th column. So let's just let's break this down a little bit and see what it is that's not working for you. If you can point out to me in the comments what isn't working, that would be quite useful. So for me, what this command is doing, so tail -n plus 2 example CSV is giving me every line from my CSV file after the second line, from the second line to the end of the file. So that's what the tail is doing. And then we're taking that output stream from that command and feeding it as input to sort. And with sort, we are doing a general numerical sort. So that should understand the numbers that have E in them. And we're using comma as a separator because it's a CSV file and we're sorting on the 7th column. It sorts as if the E minus X would not be there. So where do the E minus X numbers end up? Do they end up at the top or the bottom? So if we pipe this into less, this works for me because this number, E, which is 3 times E to the minus 299, is smaller than this number, which is E to the minus 286, which is smaller than this. So is this sorting by 3.5 rather than 3.5 times 10 to the minus 299? I think this is random, right? Okay. I think this is something which is potentially better taken offline. If you could send me this at the side of first 10 lines of the sorted output, then I can try and figure out what's going on. Because it's very odd that that's not working. I'm not entirely sure why that wouldn't be working. So it might be useful to take this offline and try and figure it out that way. Because without an example of what exactly is happening, it's not clear to me how to fix it, I'm afraid. Okay, sorry, distraction slightly, but it's always nice to try and sort people's problems out as much as we can. But okay, let's move on to grep, which is the idea behind today's lesson. So grep is what's called a regular expression program. So it's a program for searching for particular strings within a file. This can be useful in lots of ways because quite often when you've got a large file like we've got with our example.csv here, only certain lines of that file might be interesting to you. So it might be that you want to look at the expression of a particular gene or a particular set of genes, and you want ways in which you can very quickly pull those results out. And it might be that you want to feed those results into other tools. So you might want the results at the command line so that you can feed them on into other tools. And grep is a way of enabling you to pull out those particular lines out of a file very quickly. So to show you how it works, I'll give you an idea of the general pattern of the way that grep works, and then we'll try and move on to some more complicated examples. Okay, so grep is the command, grep, and the general pattern for grep is that it takes 2 arguments. So the first argument is a pattern that you want to search for. So I'm going to search for ENSG 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 3, which is the smallest ensemble gene ID that exists for humans as far as I know. So that's 10 zeros and a 3 because there's 11 digits after the ENSG in an ensemble gene ID. Okay, and then we want to search in our example.csv. So that's our comma separated value file. And what grep is going to do is it's going to look at all of the lines in that file, and it's going to return all of the lines which match our pattern, where the pattern is the first argument that we give to grep, and then the second argument is the file that we want to search. So we're going to grep, so search in example.csv for occurrences of this particular string. So an ENSG, then 10 zeros and a 3. And hopefully we're going to find one instance of that in our file. Okay, so there we go. That's the results of grep. That's what grep gives us back. And in this case, thankfully I was right. There is one instance of that ensemble gene ID in the file. So this has returned me the results, the specific results for that particular gene in this example.csv file. So ENSG 00000003. Okay, so there's the results for that particular ensemble gene ID. So this is, and the match, so the match between what I gave as my pattern to grep and what it's found in the file, that match is highlighted by default. If you're working on a system other than Ubuntu, so for example, if you're running the terminal in OS X rather in Mac OS rather than in Ubuntu, which I know some people are, then if you want the colors in your output, then you need to run grep with -- color to get it. It won't change the behavior of my grep because it does it by default. Okay, so that is an example giving us one particular gene ID as a result from my search. So that's a useful thing to be able to do if you want to be able to track down particular gene IDs. It may be that you're interested in all of the gene IDs in a particular group. So it might be that all of the gene IDs with, say, instead of having just one digit at the end, maybe you want all of the ones with 2 digits at the end. So there's a maximum of 100 gene IDs that have 8 zeros in their ensemble gene ID. So you can get ENSG 10 zeros 1 through to ensemble gene ID 9 zeros 99, and there's 100 possibilities, or 100 or so possibilities there. Anything which has an ensemble gene ID greater than 99 won't match this pattern, because instead of having a 0 in that 9th place, in that 9th number, or is it 8th number? 8, 9, 10, yeah, 9th number. So instead of having a 0 there, it will have something else. Or instead of having a 0 there, it will have something else. So this will only match a certain range of ensemble gene IDs, and it will give me all of those results for which match that. So this will give me more than one result. In fact, it's only given me one result. That's a bit of a surprise. I hate when I get surprised. I expected there to be more than that. Let's just remove one more 0 and then see. There you go. There's a few more results. So there you go. There's, of the 1,000 possible results that would have 8 leading zeros, that's the ones that we can find in this file. And if I removed another 0, I would get more results. So there you go. In this case, we can see we've got 1, 2, 3, 4, 5 genes which match that particular pattern. Okay, so that's useful, and there's nothing necessarily that these genes have in common other than the fact that they have a small ensemble gene ID number. That doesn't necessarily mean anything in terms of their function or anything like that, but we can use this to illustrate some other functions of grep. So the man page for grep is, as you would expect, extensive. It can be an extremely complicated tool, but it can be an extremely useful tool if you get to know how it works. So, man grep, you'll find is a very extensive man page. So this is very long, but there's lots of things that you can do with grep, which can be quite interesting. So, for instance, if you got lots of results from a particular search and you wanted to know exactly how many, then you can use grep to not just to tell you what the results are, but to count the number of results. So you can use -c, and that will count the number of matches that there are for this particular pattern. Actually, it won't count the number of matches, it will count the number of lines that match that pattern. So if you have multiple matches on one line, it will only count those as one match. And that's, but in its default operation, grep will match lines, not instances of your pattern. So if you have multiple instances on the same line, that line will only be matched once. So here we're going to count the instances, or the lines that contain instances of our pattern with -c, and what I said before is correct. There are 5 matches for this particular pattern. You can also start to build up patterns to be slightly more complicated by using special characters. So if you thought that this ENSG ID might appear somewhere else in the line, but what you're interested in is just the occurrences at the beginning of the line, then you can add a special character in to make sure that your matches only occur at the beginning of the line. Once you start to add special characters into any command in Bash, it makes sense to put things in single quotes. So I'm going to start to put my pattern in single quotes now, and I'm going to keep that for all of the patterns that we write from now on. So I will always put my pattern in single quotes, and anything that's in single quotes at the command line in Bash doesn't get interpreted by your shell. So if there are special characters in there, like greater than symbols, for instance, they won't get interpreted by the shell, and they won't. So if I had a greater than in my pattern, which if we look for, we'll do something else in a second. I'll show you what I mean in a second. So we'll do, if we want our result, we want to make sure that our results are at the beginning of the line, then we precede our pattern with this caret symbol, the little hat symbol, which is above the 6 on a standard UK layout keyboard. So Shift and 6 to get the little caret symbol. And this will ensure that all of your results only occur at the beginning of the line. So that's not particularly useful in the case of this grep, but if we go to, let's go back up and go to week 1 here, and let's grep for ID in q9by.txt. Okay, so ID should be the 2 characters at the beginning of the line that tells us the identifier for this protein. So we're going to grep. I'm going to put the single quotes back in, because I said I would put them around all my patterns from now on. So there you go. So ID is probably going to occur in the ID line, but it may occur elsewhere in this file, and I don't know for sure before running this whether that's the case or not. But let's run the grep and see if it occurs once or more than once. Okay, so it does occur in lots of other places as well. So you can see that we've got our ID line at the top. That's the one that's the match that we wanted. That's the thing that I was interested in. But we've also got a whole load of other lines in our file that contain our pattern, that contain ID in capitals. So that's interesting to note. So for instance, there's ID as a pair of amino acids in some of the sequence, for instance. So if what I was trying to do with this grep was recover the ID line, the identifier line from my Uniprot entry, then this grep wouldn't work. And in order to just get the ID line, I would have to modify this to ensure that ID is only, I'm only interested in instances of ID where it's at the beginning of the line. And that's what the little caret symbol means, that little hat. And so that grep will only return me the ID line from the Uniprot entry, which is presumably what I had intended to do with that grep. So The other thing I wanted to do at this point, and I can do this with the Uniprot entry from last week, with the fast2a file that I downloaded, is show you a relatively common issue that we can have with grep. So you quite often use grep for counting the number of sequences that occur in a fast2a file, right? And you do this by counting the lines which have the greater than symbol in, because the fast2a format is that so sequence IDs start with a greater than, and then you get the identifier on the rest of that line, and then the sequence happens on all of the lines underneath that, and then your next sequence starts with a greater than symbol, and so on. And so in a multi-sequence fast2a file, you will find that you will have lots of occurrences of this greater than symbol, and each one is the beginning of a new sequence. So you can count the number of sequences in your fast2a file by counting the occurrences of the greater than symbol. So in this case, there's just one sequence in our fast2a file. It's a very common thing for people to forget their single quotes or forget their quotes. There's a little bit of discussion in the comments going on about whether double and single quotes work interchangeably. They do behave differently in Bash, and I can't remember exactly what the difference is, but for whether characters that are meaningful to Bash get interpreted or not, there shouldn't be any difference in behavior. I think it's to do with variables, which is something that we'll talk about in a future lesson. So single or double quotes, in the case of what we're doing now, are interchangeable. You can use either. Okay, so if we forget our quotes, now, do we remember what the greater than symbol does? The greater than symbol is for output redirection. It takes the output of our command and redirects it into a file. So when Bash sees this command, it sees, okay, I'm going to run grep, and my option for grep is -c, but then, oh, I've got no input. All I'm doing is I'm going to redirect the output from this command, which will fail because it's got no input, into this file. And this file is actually your input file, but in this command, without the single quotes, that file is becoming our output file. So instead of it being the input file, now we're redirecting the output of our failed grep command into that fast2a file. So if we run this command, we might get a warning about us not giving a file to grep, but that will be printed to standard error, and the standard output of grep will be redirected into q9byf1.fasta, which means that file, because it's not a double arrow, we're not appending to it, we're overwriting it, and it means that file will after we run this command, be empty. So let's run the command and show that. So we get an error saying, oh, grep hasn't worked, and grep hasn't worked because we haven't given it a pattern or a file, so grep doesn't know what to do, but it's printed that error message to standard error. Its standard output has been redirected, and its standard output has been redirected to q9byf1.fasta. So now if we cat that file, we will find that it is empty because we've managed to overwrite it with the non-existent results of grep. And that's why those single quotes can be really important, because if you forget them, then you can end up doing things like this. And I have done this, believe me, I have done this, and I've done it several times, but only once have I done it on a file which I absolutely couldn't replace. And when I made that mistake, I've not made that mistake again, because that can be a fairly fundamental mistake to make. With this particular fast2a file, we can just go back to Uniprot and download it again. But if it contained the results of an experiment, which we didn't have a backup of anywhere, then we would be in serious trouble at this point. So it makes really good sense to get used to wrapping your pattern in quotes for exactly this kind of situation. Okay, so that's my word of warning for grep. Let's go back to our week 2 directory. week 2. Okay, so so we've looked at the basic operation of grep and a couple of the options that we can give grep, and there are lots of other options. So if we go back to our grep for this ENSG, we can tell it to include the line numbers, so which lines match our pattern, and it will tell us which lines our matches are on. So lines 2, 3, 4, 5, and 6, which is telling for a beginning is telling me that my my ensemble gene IDs in my file are in numerical order, which is an interesting thing to know. It's not particularly useful, but there you go. So that'll tell me the line numbers of my matches. That's quite a useful option. In some circumstances, it can be useful not just to get the line that matches, but also the lines before and after the matches. And so dash A and dash B, capital A and capital B, will give you the number of lines that you determine before and after your match. So in this case, I'm asking grep to give me one line before and one line after each of my matches, and this will be slightly complicated because those matches are all on consecutive lines. But what I end up with here is the header line, because that's the line before my first match, and ENSG 0001036, because that's the line after my last match. So it kind of expands my matches by the line before and the line after, that's useful information. The other thing that can be quite useful is to do, rather than doing, so bash by default, grep by default, like everything in bash, is case sensitive. So it will only make matches that exactly match the text that I type in. If you want to find a case insensitive match, then you can use -i, and that will make your match case insensitive. In this case, it won't make any difference to the results of our grep because we've only got capital ENSG in our file. But if, for instance, I was to want to search for the string ENSG at the beginning of the line, then by default, without the -i, this would fail to match the headline. So I'm just going to pipe the results of this into head. So this will fail to match the headline, and you know, if you pipe the results into a new command, you've lost the colouring in, and there's not really a way of getting that back. But this emits the headline. But if I do it with -i, if I do it in a case insensitive way, then I will get back that headline, because that headline begins with ENSG. It's just a lowercase ENSG. So grep with the -i gives me a case insensitive match. So there are lots and lots of options for grep. Under these circumstances, grep, for instance, might be useful for finding all of the members of a gene family. So if you're interested in the septins, then you can grep for the text sept in your file, and you will find all of the results for septin genes in your file. So there we go. I've got septin 3, septin 4, and so on. Maybe I'm interested in the septins, but I'm not interested in this antisense gene here. So septin 7, antisense 1. In which case, I can restrict my match. Now, this is where grep starts to get slightly more complicated, because in order to, if I want to match the members of the septin gene family, but I just want septin followed by numbers, I don't want the antisense ones, I don't want these P ones, so I'm not sure what the P2 and the P19 are, but maybe they're not core members of my gene family I'm interested in. So I just want sept followed by some numbers and nothing else. In order to do that, I need to use what's called a regular expression. Regular expressions we define using a programming language called Perl. And to switch that on, we need to use this -P option for Perl. So this switches on our ability to use a regular expression. And then if we want to match septin followed by numbers, then we need to say, okay, I want to find sept, and then I want a number afterwards. And you need a generic, what's called a character class. So this is a way of matching not to a specific character, but to a type of character. And they're usually, what you usually find is that a character class begins with a backslash and then has a letter after it. So \D means match any digit. So this will match the text sept followed by any digit. Okay, and this will match any identifier which matches that particular text. So if we run that, I think that this will still match all of the same things as we matched before, but this time the first number after sept will be coloured in. Okay, so we're going to build up to just matching the things we actually want. Okay, so there you go. This is matching all of the same things as before, but this time it's highlighted the sept and it's highlighted the first number after the sept because that's part of our pattern. The ones we actually want to match, the ones without the P2 and the AS1 and things like that, they might have one or more than one number. So if we restrict it just to these ones with just one number, we're going to miss some. So we need to expand this pattern. And so we want things which match sept and then one or more numbers. And to do that, we can add a plus, and this means, the plus means match the preceding character one or more times. If you used a star, it would be 0 or more times, but plus is 1 or more times. So this will give me sept plus then one or more number, and this will still match everything that was matched before, because it will still match, this pattern will still match this sept7 AS1, for example, here, because this consists of sept and then one or more numbers. So it will still match. The way to restrict this now to just the ones that we're interested in, so to just the septins without the P's or the AS's or anything like that, is to restrict this match to being at the end of the line. So to say, well, we want this, we're interested in this particular pattern and we want to match that pattern, but we only want to see that pattern at the end of the line. Okay, so we go back to the pattern that we've just done, and in order to do that, it's like putting the hat symbol at the beginning of our pattern, which says only match this pattern if it occurs at the beginning of the line. The way that we do it at the end of the line is at the end of our pattern we put the dollar symbol, and the dollar symbol says only match this pattern if it occurs at the end of a line, and this will eliminate those matches that we're not interested in. Okay, so now we've got rid of the sept14 P19 and the sept7 AS1 that we had before. They're no longer included in our matches because we're enforcing the fact that this match occurs at the end of the line. And so that's beginning to unlock the power of grep to do lots of really interesting things. And the best way of unlocking that power, despite what Peter says in the comments, is to learn how Perl regular expressions work. And I realised that they can begin to look really, really ugly and complicated, but they can be extremely powerful for finding the results that you want from this kind of search. Okay, I think I might talk a little bit more about grep tomorrow. I'll try and formulate some more interesting things that we can do with grep. Maybe spend another 15-20 minutes talking about it tomorrow to try and show you a bit more of the utility of it before moving on to the next thing. But suffice to say, we're only beginning to really scratch the surface of what grep is capable of doing. and I could spend days and days talking about it, but I'm not going to, don't worry. Okay, so with that, I'm going to sign off for today. So thanks again for dropping by. Thanks for your attention. I'm going to finish by going back to Jacob's picture of the day, because I think it's a nice message to finish on. So that's going to be the last thing that we'll have on the screen for today. Okay, so again, thanks for dropping by, everybody. Stay well, keep safe, and I will see you all tomorrow for a bit more grep and some other things besides. So see you tomorrow, everybody. Bye.